In the lab, remember, this week's lab is due on Friday. Some people have already turned it in early, and I'm seeing some common mistakes. So when we did this experiment, where we're looking at the salt-starch mixture in the cell to see if the salt came out, or if the starch came out, or if the water went in, or if the iodine went in. There's a series of questions afterwards that said, hey, what was your hypothesis and was it correct? That part's pretty straightforward, right? You just say, my hypothesis was this, it was correct. Then it asks, like, for example, how do you know if water went into the cell? If it gained weight, water went into the cell. How do you know if iodine went into the cell? The inside of the cell changed color, iodine went in the cell. How do you know if salt came out of the cell? If salt came out of the cell, then it would turn cloudy from the silver nitrate. How do you know if iodine left the cell? Because if iodine left the cell, then the water in the beaker would have turned purple. That's what those questions are asking. So far, most of the people who've turned it in, I don't think quite understood the questions, which is like not their fault. Sometimes it's hard to understand what the question's asking. So that's what the question is asking. So you guys know. All right, let's start some attendance. Boggs is here. Harrison is here. Vincent is here. Kesterson is here. And Morrison is here. Online we have Aki, James, and King. And again, don't forget, if you're online, you need to send the attendance words to be considered present. I got one of this. James. What? Oh, yeah. So Ward is here in red, and see that Harrison is also in red. Oh, and Hanson's also in red. Ward. Green is here in red. So another thing about independent work. A classmate inspired me for this. This is, I think, a really good idea. If you're going to share something with me, which I love, like you found you can't stumble upon something about biology, might as well get some points for it. So like if this person found a cool article about how enzymes are used in like cleaning materials and just wanted to share it with me. And then I got back and they got a point. It's like, so when you want to share something with me, you can say, hey, I was wondering about this, and then I found the answer. I might as well take advantage of that. Get some points. Don't forget, if you do just share something with me for the sake of like, I had this question, I looked it up, and this is the answer. And remember, you don't have to worry about plagiarism. You don't have to worry about putting it in a Word document or a Google document. You just send me the email, and in the email say, I had this question, I looked it up, according to this source, here's the answer, and then automatic 1 point. Unless, of course, you just completely misunderstood what the website said, which so far hasn't happened. At least not with a simple question. I've had a lot of people write papers where they'll misquote their source, but not just like one answer to a question. Anyway, less than a minute left. Morning. Linda Q is online. Dawson? Yep. Dawson is here. Paula is here. All right, everybody's accounted for. It's 8 o'clock. Let's get this party started. So for those of you who came in, well, on time, but just now, I gave a little information about the lab. So if you get a chance, when I post this video, you might look at that before you turn your lab in. It might give you some helpful advice. Anyway, let's jump into it really quickly one more time for the sake of recording. Aki is online. Boggs is here. Bonner is not here. Clay is not here. Dawson is here. Ellis is not here. Paula is online. Green is here. Harrison is here. Henson is here. James is online. Kesterson is here. King is online. Lewis is not here. Linda Q is online. Morrison is here. First name Grace is not here. Ward is here. Wilcox is not here. Wilder is not here. Parks is not here. So we're done with attendance. Let's jump into it. Try to get caught up a little bit. I'm trying to move a little bit quickly. So this is relatively important. Last thing we were talking about was enzymes. Then we talked about enzyme inhibitors, which are things that stop an enzyme from doing what it's supposed to do. Which, now that I think about it, I didn't say this. So before I move forward, let me step back for a second. I explained how enzyme inhibitors work, right? I explained that they basically work by changing the active site, either by blocking it or changing the shape. Either way, they keep the substrate from coming in and attaching to the enzyme, therefore they keep it from working. What I didn't explain was why this happens. So you can think of your cells as little bitty factories, right? And these proteins inside your cells, these enzymes, are smaller little things inside the factory. Each little enzyme is trying to make a product, right? And your cell does not always need all the products all the time, right? Sometimes it needs particular products, sometimes it doesn't. And that's where those things come in. Even though I'm not going to ask you on the exam, that's why we have enzyme inhibitors. And I use the analogy, do you guys remember fidget spinners? You old enough to remember those things? Remember they were like a huge trays for like a year or two. Like every little kid had them. Matter of fact, you guys might be the right age to where you were little kids at that time. Anyway, so imagine if you owned a toy factory and it's Christmas time, and this is back when fidget spinners were a thing. You would say, all right, let's get some more of these machines in here. Let's get some more people and more resources making fidget spinners. That's going to be our bread and butter this summer or this winter, right? So you spend a lot of resources making fidget spinners. And then years later, it's not as popular, so now you don't need as many fidget spinners, right? So you put your resources towards building other stuff. Same thing in the cell, right? So your cell needs things sometimes and needs those enzymes to produce those things, but then sometimes it doesn't, so therefore it needs a way of turning off those enzymes, whether it be temporarily or permanently, and that's where those enzyme inhibitors come from. So there we go, now we're caught up. Now let's move forward and talk about some other things. Enzymes often require helper molecules, so even though I was just showing you all these examples of this one enzyme attaching to this one substrate, and then we get a product, it's not always like that, right? Sometimes we need these helper molecules. They can form temporary or permanent bonds. As far as the exam is concerned, this information I'm about to give you is probably a little bit less important. I don't think I'm going to go this deep on the exam. We will be talking about some cofactors and some coenzymes later in the semester. So for that reason, it is important for me to introduce them now so you know what I'm talking about later. So when I throw out the word coenzyme, you're not like, what in the world is coenzyme? It's here I am telling you. So first of all, let's talk about cofactors. Cofactors are inorganic ions, right? So magnesium or iron is a great example of that, right? So hemoglobin, right? The stuff in your blood that carries oxygen, it needs iron, right? So that's a great example of a cofactor. Which, by the way, notice I only listed about 2 examples. This is a great independent work topic. So you could just send me an email for 1 point, say, I wanted to know what the rest of the cofactors were, and I looked them up, and according to this source, blah, 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 right? Just send it to me. Or if you wanted to, you could write a whole paper about it and talk about what iron does and what magnesium does and what the other ones do. It's up to you. Anyway, the next one is called coenzymes. These are actually organic molecules, and they're usually, which means they are carbon and hydrogen based, right? So we've got cofactors and coenzymes, both of which help enzymes do their jobs. Sometimes they attach permanently, sometimes they attach temporarily. And again, the reason I'm really focusing on this is because later in the semester, I will bring these words up again, and I don't want you to be confused when I bring them up. So both of these things participate in reactions without being altered. So they are also recycled and reused, right? So very much just like the enzyme itself. Remember, enzymes don't get used up in reactions. They just get recycled and reused. And as a little side note, vitamins often serve as the source of coenzymes. So that's another great independent work topic. Whether you want to write a paper about it or just a simple answer, right? So what are the vitamins that serve as the source of coenzymes? And again, if you wanted to write a paper, you could get a little bit deeper. So like, let's say it's vitamin C. Which enzyme does vitamin C work with? How does it work with it? You know, you can dive deep if you want. Anyway, speaking of which, the first word for attendance, right there on the board. I'm underlining it. I'm not even going to say it. So there it is. That's, whoa, it didn't work. It shows up on my screen. Let's try this. There we go. What? Are you kidding me? Anyway, that's the first word for attendance. Vitamins. I guess I'll just say it since it's one we underline it. Vitamins is the first word for attendance. And again, if you're online, I'll remind you, you have to send those words in to get to be counted as present and to get your extra credit points. Anyway, any questions about this slide? Before I actually go back and start to talk about something I was talking about before. My looks like my PowerPoint slides are a little bit out of whack. So that being said, so again, I was talking about why we have inhibition, and I forgot that I actually had it on my PowerPoint. This is a relatively new addition to my PowerPoint, so I forgot about it. So feedback inhibition is the use of the product of whatever chemical reaction we're talking about to regulate its own further production. So this is exactly what I was mentioning when we first started the lecture today. Completely forgetting that it was coming up. So here we go. Here's a great example. Here's this enzyme. You know, I call it the Pac-Man enzyme. Why not? It looks like Pac-Man. There's the substrate, right? The substrate comes into the enzyme. We have another intermediate substrate. I don't like that picture. This is from your textbook. And that really makes it more complicated than it needs to be. Basically, what we're showing here is once this chemical reaction happens, then we finally end up with the product, which in this case is a yellow square. But that yellow square, which is the end product, also serves as the enzyme inhibitor, which is, again, it's a great way of slowing down chemical reactions or keeping chemical reactions from happening that don't need to happen. So if your cells need a yellow square and you're producing a lot of yellow squares, eventually there's going to be so many yellow squares that they're going to start inhibiting the enzymes that are making them, which is what you want, right? Because you don't want to devote all your resources to making this one product. So any questions about feedback inhibition? All right, now we're done with the background stuff, and now we can get into the more complicated stuff. As far as this exam is concerned, this is mostly what your exam is going to be about. It's going to be about 4.2, 4.3, yeah, mostly that. There will be other stuff, but most of the exam, as far as chapter 4 is concerned, is going to be talking about cellular respiration. And again, like I mentioned the other day, for this portion of the exam, most of the questions are going to be, and again, there's 3 stages of respiration, which you're about to learn. Most of the questions are going to be, in which stage does this happen? In which stage is this produced? And which stage is this used, right? So you need to know the formula. You need to know which stages uses those things, which stages produces those things. So keep that in the back of your mind as we go through this, this portion of the chapter. So here's the learning objectives for this portion. At the end, you should be able to explain how ATP is used by the cell as an energy source. And you should be able to describe the overall result of cellular respiration, or excuse me, glycolysis, in the terms of molecules produced of the breakdown of glucose by glycolysis. So of these two, I would say this one's more important as far as the exam is concerned, the second one. You should be able to describe the results of glycolysis, which is what you have to learn. But before we can talk about that, let's back it up a little bit and talk about ATP. So again, when we're talking about chemical reactions, we're saying activation, whether those chemical reactions are endergonic or exergonic, right? So whether they require input of energy or if they put energy out, either way, even if it's putting energy out, they still need that activation energy, right? All chemical reactions need that energy to at least start the reaction. And as far as we're concerned in this class, this is where that energy always comes from, ATP. ATP, and this will be a test question. and it's not written the way it is on the exam, so I'll go ahead and tell you how it is on the exam. ATP is where cells directly get their energy. So the question will be, how do cells directly get their energy, or what molecule do cells use to directly get their energy? And the answer is ATP. Make sure you know that. What is ATP? It is a small, simple molecule. I'll show you a picture so you can see, oh, sorry, there at the bottom right. It's a relatively small, it's bigger than the molecules that you guys drew in lab, but think about all the other molecules that you saw in chapter 2 when we talked about carbohydrates and lipids and proteins and nucleic acids, right? So this is relatively small. It's actually so small, speaking of which, not that you need to know this, but if you remember A from DNA, CTAG, this is the A from DNA, so it is actually nucleic acid. Well, with some extra phosphates attached to it. Anyway, it contains potential energy for cellular work. And again, like I said, it is the primary energy currency in cells. So that will be the test question. Again, I'm not sure how it's going to be written. It could say, what is the primary energy currency in cells? It could say, what molecule do cells directly use for energy? Something like that. But yeah, that's a very important little point. It powers a majority of energy requiring cellular reactions, which is a great independent work question. If I'm saying ATP powers a majority of them, what are the exceptions to the rule? So again, you can just send me an email with a short answer. According to this source, these are the other power molecules. Or if you wanted to, you can make a paper out of it and dig deeper and get more than 1 point. It's up to you. And basically what they are, and this is the best way I like to describe it, is rechargeable batteries for cells. And I'll explain that last little point as we move forward. Obviously, the battery part makes sense, right? Because I'm telling you it's like the energy currency. This is where cells get their energy. So imagine everything in your cell runs on batteries. Well, that's what ATP is. That's the batteries for all these things that run in your cells. But the part that I will have to explain later is the whole rechargeable part. So once the battery's dead, it's not just dead. You don't just throw it away. It can be recharged. And that's what we'll talk about. But to be able to explain that, we need to talk a little bit more about its structure. Right? Before we talk about the function, we'll talk about the structure, which I'm about to do on the next slide. Does anybody have any questions about this slide? All right, so again, ATP. This is an adenosine molecule, which is this thing right here, with a tail of 3 phosphate groups. I'm not going to ask you this, but to be able to just understand how ATP works, then you do need to understand this, which is why I'm explaining it right now. When it's used, right, it's broken down to something called ADP. So instead of triphosphate, it's called diphosphate, which makes sense because when it's used, when it's broken down, you're losing one of those 3 phosphate groups, and then you're down to 2. So tri means 3, di means 2, right? And when this happens, that's what releases the energy, right? So it's the release of the phosphate that makes energy available to the cells. So on this slide, this is the bullet point you might need to know for the exam. So again, you definitely need to know that ATP is what cells directly use for energy. And not only that, but you need to explain how, right? How does it work? And it works by releasing a phosphate group. Any questions about this slide before I show you the visualization of the words on this slide? All right, so here we go. There's your adenosine triphosphate, right? You got the 3 phosphate groups, and then you release energy by releasing a phosphate group. Actually, and also let me back it up a step, 2 steps maybe. Not that I'm going to test you on this, but I really want to make sure you understand. So again, none of you are chemists, but you can see this, right? These things that I'm circling. What do all these things I'm circling have in common? Yes, they're negatively charged, thank you. And another thing that I don't think I've taught you because you don't need to know is that oxygens themselves are very negative. And another thing I haven't taught you, I don't think, because you don't need to know, is that double bonds are very negative. So all those balls are very, very negative, which goes to show you, like I was talking about earlier in the week, those balls don't want to be next to each other. They're repelling each other. They're really negative. So it takes a lot of energy to squish those things together. Hence, once you release one, all the energy it took to squish those things together is then released. Not that I'm going to ask you that, but this is why ATP is such a high energy molecule. And this is why losing or releasing one of those phosphate groups releases so much energy. Anyway, back to where we were. So basically, again, ATP energizes other molecules by transferring a phosphate group to them, right? So the phosphate group just doesn't go off into the cytoplasm. It's actually transferred to another molecule. As far as I'm concerned, that's not necessarily important for the exam or the quizzes or anything. Maybe the quizzes, I don't think I'm gonna have that on the exam. So again, I want you to focus, I want you to know that ATP, once it releases one of those phosphate groups, that's where it releases the energy. But yeah, side note, it doesn't just go flying off into nowhere, right? It actually transfers that phosphate to another molecule. Here's some examples. This is some pictures from the old textbook. Here you have ATP. We're trying to move a motor protein. So we transfer a phosphate group, and then it moves. That energy is then used to move the motor protein. And then, of course, you're left with ADP and then a phosphate group. And this is just an example. If you don't, I'm not going to ask you any questions about what's on that side or this slide. We've already talked about this, right? This is active transport. So again, with active transport, when you're pushing something against this concentration gradient, that requires energy. Where does that energy come from? ATP. How does ATP give it energy? Well, it transfers one of the phosphate groups to it, and in the process of doing that, it releases energy, right? And then, of course, not that I'm going to go this deep on the exam, but then after the deed is done, right, after it's done the work that it needs to do, the energy was used to move this solute through. So then when it's done, it releases that phosphate. Then it does go into the cytoplasm. And so then floating around in the cytoplasm, you now have an ADP phosphate. And of course, eventually, we're going to put those things back together. And then once again, another example, and these are just examples, so I'm not going to ask you questions about them. But in this example, we're not moving a motor protein, we're not doing active transport, we're building a molecule. Remember, like I said earlier in the week, it takes energy to build molecules because these atoms don't want to be close to each other. So you have to push them together, so to speak. And that energy comes from ATP. Specifically, it comes from transferring the phosphate group, which releases the energy. And in this case, that energy was then used to bring X and Y together. So any questions about that example? Okay, like I said, understanding ATP as a battery, that's pretty simple. Now let's explain the whole rechargeable battery part. First of all, cells spin ATP continuously, right? So you are constantly making and using ATP. So it is recycled when ADP, right, the adenosine diphosphate, the only one with two phosphate groups, plus another phosphate group, once you combine them, you then make ATP again. So it's a pretty simple cycle. Yeah, still talking about this bullet point here. You're going to learn a lot of cycles in the next couple of chapters, and this is the easiest one. It's pretty simple, right? You have ATP up here at the top, right? That's your charged battery. It releases the phosphate group to charge whatever, give energy to whatever it's getting to. Now the dead battery is ATP. All right, now we want to recharge it. So how do we do it? We add another phosphate group to the ADP, and then we're back to ATP. Pretty simple, right? Anyway, here's another word. I'm probably not going to ask you this, but there it is. About 10 million ATPs are consumed and recycled each second in one working muscle cell. So again, independent work topic. For a simple question, you can just ask yourself, what about how many ATP are consumed and recycled each second in a brain cell or a kidney cell, whatever cell you can think of? You can look that up for an easy answer. If you want to do a short paper, go figure out, oh man, this might be an easy answer too. If we're doing about 10 million ATP in one cell, one working cell, then how many in your body, for the average human, how many ATP are you consumed each second in your entire body? That'd be interesting to know. And again, there might just be an answer, so that might be your 1 point independent work for each second. I was wondering how many ATP the human body consumes on average, and according to this source, this is the number. All right. Any questions about this? All right, here's another picture showing you the same thing. This is the picture from the old textbook. Again, here we have the dead battery, right? The ADP. We need ATP, so what do? We push that phosphate back to the ADP, and that gives us ATP. Now we have a charged battery. That charged battery then releases its energy for work, also a phosphate group. So then we're back down to the dead battery, right? It's that simple. So any questions about that? Okay, so before we get into cellular respiration, and this is important, so if you guys are working on a lab or, I don't know, playing on social media, you might pay attention to this, because this is not in your textbook. Your textbook is flawed, in my opinion, because before going into the details, I feel like a good broad overview would be important. So before we go into the specific stages and give you all the information, I want to give you a broad overview. So here we go. Cellular respiration consists of 3 main stages, and you need to know this. Glycolysis, citric acid cycle, and electron transport. You don't need to memorize that because most of these questions, you need to know it, but you don't need to memorize it. What's going to happen is for your exam, most of the questions, the choices in the questions are going to be this. Because again, I might say, in which stage is oxygen used? And you'll need to know that's electron transport. In which stage is glucose used? And you'll need to know it's glycolysis. In which stage is carbon dioxide produced? And you'll need to know it's the citric acid cycle. So again, here we go. We're going to go do the overview, which is not in the textbook, and then we'll get into some more details, which is in the textbook.
I have a question.
Yes.
ADP's main function is just to recycle it back to ATP?
Yeah, in a sense, ADP doesn't have a function. In a sense, it's just the dead battery, if you look at it that way. Which is another good independent work topic if you want to look into that. So as far as we're concerned, the ADP is useless. We just need to squish another phosphate group onto it, and then we can use it. So then the question could be for independent work. Does ADP have any other purposes? And then what about AMP? What do you think AMP might stand for?
Oh, mono.
Yeah, adenosine monophosphate. Actually, adenosine monophosphate. Anyway, yeah. And adenosine with only one phosphate group. Things you could look into if you want. So anyway, you download the PowerPoint. You can watch this little animation. This shows you some cellular respiration work. Here we go. This is a big overview, so I'm going to go through it, not quickly, but fairly quickly, because I'm just giving you the overview. Later, when I'm done with the overview, we'll go in and talk about this in more detail. So, stage 1, glycolysis. This is when glucose is split into 2 molecules of something called pyruvate. And this is usually located in the cytoplasm. So for the exam, here's what you need to know. When is glucose used? Glycolysis, right? When is pyruvate produced? Glycolysis. Which of the 3 stages happens in the cytoplasm? As in not in the mitochondria? Glycolysis. I will say, point this out. In your textbook, I think, they call pyruvate, another word for pyruvate is pyruvic acid. So if you're taking notes, you might want to write that down. So when I say pyruvate, I also mean something called pyruvic acid. Reason, well, I'll tell you why later. I don't like to call it pyruvic acid, and you'll see why later. It's actually to make your exam easier. So yes, pyruvate is what we, is also known as pyruvic acid, but we call it pyruvate. Also, I put a star next to the word usually, because this is not always the case, but as far as we are concerned, like as far as studying is concerned, and you're getting ready for the exam, getting ready for quizzes, you can even get rid of the word usually, right? As far as we're concerned, glycolysis always happens on the cytoplasm. So any questions? And again, we're going to come back to this. If there's any questions so far? All right, so that's really a broad overview of the first step. The second step is called citric acid cycle. It's also called, this is another AKA, Krebs cycle. But, so when you're reading in the textbook, I think they call it the Krebs cycle. I don't call it that. And there's a particular reason why, and you'll see why here in a second. So the citric acid cycle, again, the second stage of cellular respiration. This does happen in the mitochondria. So well, that's a good test question. Which of the 3 stages happens inside the mitochondria? Well, this is one of them. And the next one too, but yeah, this happens inside the mitochondria. This is when we've completely broken down, I should reword that. Complete breakdown, yeah, sort of. I don't want this to confuse you. Here's the more important part. Focus on the CO2 part. Citric acid is where we produce carbon dioxide. So again, we had that pyruvate. Remember, pyruvate was the product of glycolysis. Then we take that pyruvate, we do some things that I'll tell you about later. That then comes into citric acid to the citric acid cycle, and we break it down even more. And that is when we produce carbon dioxide. So that will be a test question. And which of the three stages, is carbon dioxide produced? And the answer is the citric acid cycle. And that is why I don't call it the Krebs cycle, because it's a built-in hint. The citric acid cycle is where we produce carbon dioxide, all those C's, right? So you get any question on this exam that asks you anything about carbon dioxide, like when is it produced, in this case, or if it's photosynthesis, when is it used? And the answer is always going to be the cycle, right? And here in respiration, citric acid cycle produces carbon dioxide. And then there's another cycle of photosynthesis that uses it. So again, for the exam, there's a question that ever asks when was carbon dioxide produced or used, the answer is always going to be some sort of cycle. And again, we're going to talk more about the citric acid cycle later. We'll get into more details, but for now, this is just an overview. So any questions about that? All right. So glycolysis and the citric acid cycle generate a little bit of ATP directly, right? Just a little bit. As a matter of fact, your textbook will give you some numbers, and I'll give you some numbers later. And I'm not really too interested in those numbers, but just know that there's a little bit produced. Because remember, this whole thing is about producing ATP, right? That's the whole main goal of cellular respiration, is to produce ATP. So obviously, we need to discuss which ones produce how much. So again, the first two produce a little bit, but they produce a lot more indirectly by transferring electrons from the fuel molecules to a molecule called NAD+. If you're taking notes, I wouldn't even worry about this bullet point yet, because we're going to come back and talk about it later. But for now, again, just to introduce you to it, whenever you see any molecule that looks like this, there's three of them that I'm going to teach you about. This is the first one. And they all kind of look like that. NAD+, FAD+, there's some other, there's another one, and again, we'll cross that bridge when we get there. But if it looks like that, you should think to yourself, oh yeah, that's the thing that carries electrons. And for me, when I was a student, what helped me remember that is, does anybody remember what charge does an electron have?
Negative.
Negative, thank you. So obviously, it's going to be very attracted to this positive thing, right? They're going to come together. So that's how I always remembered it. So again, I'm going to come back to this molecule later and some different versions of it. But for now, you just need to know, again, the first two stages produce a little bit of ATP. But the most important thing that they're doing, and we're going to talk about this again later, is they are transferring high energy electrons, which is where this molecule comes in. So it's the electron transfer that forms molecules called NADH. Excuse me. Let me back it up again. So again, this thing, that thing's the thing that's going to grab the electrons and transfer them somewhere, right? And then technically, just to make things more difficult, technically once it has those electrons, it's a different name of the molecule. Once it gets those electrons, it is then called NADH. For the exam, I'm not going to have you differentiate between NAD+ and NADH. Once again, when you see this, or anything that looks like that, your brain should just think, that's the thing that's transferring electrons. Because again, for this 100 level non-biology major class, I'm not gonna have to differentiate between what it's called before it gets its electrons and what it's called after it gets its electrons. Of course, it would help knowing the difference, because in the test, I will use these words in the questions, like, hey, which stage uses NADH to carry this? Or which stage, I'll leave it at that for now. Point is, when you see something like that, you should think it transfers electrons. And I'm not going to get so specific where you have to know the difference between the two. But again, if you did need to know the difference between the two, obviously the one with the plus is the thing that's ready to accept an electron. The electrons are negative, they're going to go together. This NADH, it's not ready to accept an electron. Anyway, any questions about that? And again, we're going to come back to that. All right, so where are these electrons going? The meat and the potatoes of it, in my opinion, the electron transport chain. So this is the big one. Of the three, this is the big one. This is the last one of the three. Your textbook also calls it oxidative phosphorylation. But again, I just like electron transport chain. Depending on the source, you could get either, they could describe it either way. Same name, or same thing, different names. So what happens in the electron transport chain? these electrons are stripped of their energy. So remember, for the first two stages, we're just stripping away high energy electrons. And I'll explain what high energy means later. And then those high energy electrons come to the transport chain. And once they get there, like I said, they're high energy, right? So we're stripping some of that electron away. So the electron transport chain has proteins that are embedded in the inner membrane of the mitochondria. This is another concept we're going to come back and talk about in more detail. I'm going to show you pictures and thoroughly explain it. But for now, this is just an overview, just an introduction. And then finally, the electron transport from that NADH, which is the thing that's carrying those high energy electrons, to oxygen releases a lot of energy, and that energy is used to make most of the ATP. There's a lot of test questions here. So, of the three stages, which one uses high energy electrons, right? It's electron transport chain. Hopefully that's easy to remember, it's in the name, which is why I don't call it oxidative phosphorylation, right? Electron transport chain, hopefully that's easy to remember for the test. Which one uses high energy electrons? Electron transport chain. Which one uses oxygen? Electron transport chain. Which one produces water? Electron transport chain. Which one produces the most ATP, right? All three of them produce ATP, but this one produces the most. So again, when you think about it, the other two are there for this one, right? The other two are just there to strip away high energy electrons, right? To take that glucose or whatever other molecule and break it down, and in the process of breaking it down, they're also stripping away those high energy electrons. They can be used here where we make most of the ATP. So once again, this is an overview. We'll come back and talk about this in more detail. Speaking of an overview, you definitely need to know this. The overall equation, glucose plus oxygen gives you carbon dioxide and water and about 38 ATP, or 32, depending on your source. So you definitely need to know that. I would study that, memorize that before you memorize anything else. You should know that we use glucose and oxygen. We break down glucose in the presence of oxygen, and the byproducts are carbon dioxide and water, but we're also producing what we're after, which is about 32 to 38 ATP. Know that first. Then once you have that memorized, then memorize the three different stages, right? Glycolysis, citric acid cycle, electron transport chain. And once you have those things memorized, then you can start putting them together, right? So in which stage is glucose used? Glycolysis. In which stage is oxygen produced? Electron transport chain. In which stage is carbon dioxide produced? Citric acid cycle. In which stage is water produced? Electron transport chain. And in which stage is ATP produced? All of them, especially the last one. So any questions about this slide? And again, we're going to come back to all this and talk about it in greater detail. All right, here we go. This is a picture not from your textbook. This is from the old textbook, but I like it. It's another great thing to study. It has a lot of test questions on here. So excuse me for repeating myself, but here we go again. Which stage uses glucose? Glycolysis. Which stage produces pyruvate, aka pyruvic acid? Glycolysis. Which stage happens in the cytoplasm, as in not inside the mitochondria? Cytoplasm. Which stage happens inside the mitochondria? Citric acid cycle and electron transport chain. Which stage, at least in this picture, which stage produces ATP? All of them. Which stage produces the most ATP? Electron transport chain. And then finally, which stage uses high energy electrons? Electron transport chain. So a lot of test questions in that one picture. Any questions about that one picture? All right. So now that we're done with the big picture stuff, right? We're done with the overview, the introduction. Let's go back and look at it again, but this time in more detail. Here we go. Glycolysis. Back to the beginning. Stage one is glycolysis. Again, like I already said, like you already know, this is the stage that uses glucose. None of the other stages directly use glucose. Because in glycolysis, glucose is broken down into, like we already said earlier, pyruvate. So let's quickly talk about how that happens and give you some reminders too. Glucose is a 6-carbon molecule, not that I'm going to test you on that. but that helps to keep track of what kind of molecule we're dealing with here. It's a 6-carbon molecule, and if you split something, a 6-carbon molecule in half, right, completely in half, then you're gonna be left with 2 molecules. How many carbons would each of those molecules have? Of course, it's right there. 3, right? So if you have one 6-carbon molecule, I'll just try to draw it. Say that's, of course, that looks like it's an oxygen. But I'm drawing those as little, maybe it would have been better if I just drew the C. But anyway, 1, 2, 3, 4, 5, there we go. So 6-carbon molecule, if you were to break it in half, you would have 2 3-carbon molecules, right? That's what happens. So again, we break down glucose, we produce pyruvate. Each of those 2 pyruvates has 3 carbon molecules. Again, I'm not going to ask you that, so I might as well just X through that as a remind you. I'm not going to ask you about how many carbons they have. In the process of doing that, this is the more important part, again, we're stripping away high energy electrons. So there's high energy electrons in these bonds that make up that glucose. And when we break it down, we can strip it away. And again, as a reminder, it's this molecule that's doing it. The NAD plus strips it away, and then technically once it has the electron, it is then called NADH. And then that NADH can then leave and go to the electron transport chain and say, hey, here's some electrons for you. Do your thing. So again, like all 3 stages produce some ATP. Glycolysis does not produce much. Specifically, it yields a net gain of 2 ATP molecules per glucose molecule. And you do not need to know that. We're going to talk about it a little bit later again. But the specific number of 2 ATP, well, I don't want to say you don't need to know that, because maybe I'll have an extra credit question, but most likely I'm not going to ask you specifically how many ATP are produced in glycolysis. What you do need to know is glycolysis and citric acid cycle produce a little bit of ATP. So again, glycolysis, one of 3 stages of cellular respiration. Now to make it even more complicated, glycolysis itself is broken down into 2 phases. I'm going to talk about them because your textbook does, and I guess it's good to know it, but I'm not going to ask you any questions where you have to differentiate between the 2 phases, at least not in the exam. Maybe in the quiz, but not in the exam. So the 2 phases of glycolysis are energy investment and energy harvest, which makes sense because if you're a thinker, hopefully when you read this bullet point, you were like, wait a minute, what does he mean by it yields a net gain of ATP? Why not just say it makes 2 ATP? What is this net gain? So we'll explain that here in a second. So once again, as a reminder, glycolysis is the one of the 3 stages that happens outside of the mitochondria in the cytoplasm. Again, mitochondria is the one stage that starts with glucose. Again, we break down the glucose to produce 2 molecules of pyruvate. So again, this is the only stage that uses glucose. It's the only stage that produces pyruvate. There we go. Let's talk about the 2 different phases. First phase is the energy investment phase. So even though we're trying to make ATP, right, that's the goal here. The goal is actually to strip away high energy electrons, but we're also making a little bit of ATP. But despite the fact that we're still trying to make a little bit of ATP, we still need to put ATP into the system to get it going, right? So here we go. We spend 2 ATP right off the get-go in the energy investment phase. So once again, for the exam, you don't need to know this, but it's in the textbook, so I'm explaining it. So again, what we have, remember, let me back it up a second. Here we have these regular old pyruvates, right? These 3 carbon molecules. And what's happening is, in this energy investment phase, we're using 2 ATP to give each one of those pyruvates a phosphate group. Right? So there we go. We've invested our energy. Now we can do what we're after. We can strip away these high energy electrons. And in that case, we also add another phosphate group. That's a whole different story. It doesn't matter. Actually, none of this matters as far as the exam is concerned. Again, stripping away high energy electrons. That is the energy harvest phase. So once again, we have to put in a little bit of energy to get energy out the other. In this case, most of that is coming from high energy electrons that we're stripping away. But again, like I said earlier, we also do produce a little bit of ATP. And specifically, like I said, it's a net gain of 2. And again, where I was coming with that with the net gain is because, yes, here you can see we produced 4 ATP, right? We produced 4 ATP. However, it cost us 2 ATP to get there. So that's why we say it's a net gain of 2. So yeah, we produced 4, but it cost us 2 to get there. So in the sense that we really produced 2. So any questions about this slide, which is, as far as the exam is concerned, very unimportant. Same with the next slide. This is from the old textbook. I just thought I would keep it in here. It's just showing, in this case, when we're making ATP, there's an enzyme, and you can look it up if you want for independent work. What is the name of the enzyme the glycolysis uses to produce, to produce ATP? Yep. So again, we had that pyruvate. Remember that 3-carbon pyruvate? Then we put a phosphate group on there. Then we put another phosphate group on there. And now we have this enzyme that takes away one of those phosphate groups and attaches it to an ADP, giving us ATP. So again, as far as the exam is concerned, that's very, very unimportant. But you can look it up if you want for independent work. So any questions about glycolysis? Because now we are completely done talking about glycolysis. We're not coming back to it. Yes, sir.
So would the, is it called phosphate, right? A phosphate? Would it move the ADP, and does it just remove itself from that enzyme and can another like cell or molecule make it swap?
If I understand you correctly, what's happening is this enzyme is actually removing the phosphate from here and squishing it and adding it to that ATP, which produces the ATP.
So it's still in that slot where in the enzyme.
Oh, good question. No, if you were to see this picture would be better if it didn't have this enzyme over here empty.
Okay.
Yeah.
So then a different molecule could fit that slot.
Yeah.
Okay.
Yeah. Once, just like any other enzyme, once it's done with its job, it's ready to do it again. So its active sites, in this case, 2 active sites, would be empty again. So no more questions about glycolysis, right? We're done with the overview. We're done with the specific conversation about glycolysis. We won't be coming back to it. All right, let's talk about the other one. The other 2, citric acid cycle and oxidative phosphorylation, aka electron transport. By the end of this section, you should be able to describe the location of the citric acid cycle. That one is important. That's the mitochondria. That will be a test question. And electron transport chain in the cell. And of course, that one is also in the mitochondria. So that first bullet point, definitely a test question. 2 test questions. No, 1 test question. Which of the 3 stages happens inside the mitochondria? Citric acid cycle, electron transport chain. You should also be able to describe the overall outcome of the citric acid cycle. And you will need to know a little bit of that for the exam. I'll explain that when we get there. And the overall outcome of citric electron transport chain. And again, I'll explain that when we get there in terms of the products of each. So I've already told you the products of each, right? I've already told you, well, no, okay, I'll come back to it. Anyway, and then finally, you should be able to describe the relationships of glycolysis, citric acid cycle, and the electron transport chain in terms of their inputs and outputs. So here we go. Those are the learning objectives. Let's jump into it. Let's see. The next word for attendance will be the one I'm circling now for work. Hey, it worked. So that's the next word for attendance. All right. So again, let's take a step back and look at the big picture. We are talking about stage 2, right? We're done talking about stage 1. So to remind you, even though it's just like a minute and a half ago, the end of stage 1, what did we produce in stage 1 other than a little bit of ATP and some high energy electrons? We produced, I don't know if you're going to say it. Pyruvate? Yeah, pyruvate. So when you send me the words, again, as always, when any of you send me the words, if you answer the question in class, remind me of the question you answered. But yes, glycolysis produces pyruvate. So obviously, pyruvate is going to be the thing that goes into the citric acid cycle. But that would be too easy. So whoever was in charge of making everything decided, all right, well, there's going to be biology students one day, so let's make this more complicated. So before the pyruvate can go into the citric acid cycle, it has to be groomed. AKA converted, right? So the citric acid cycle cannot use pyruvate as is, right? So we've got to make some changes to it. Let's talk about those changes. First of all, it loses, remember pyruvate is a 3-carbon molecule. It loses one of those carbons as carbon dioxide. So once again, test question. Which of the 3 stages produces carbon dioxide? Citric acid cycle. Now I will say, I'm not going to get very specific and ask you, where does that carbon dioxide come from? You need to know it comes from the citric acid cycle, but I'm not going to require you to remember, oh yeah, well the carbon dioxide comes from that 3-carbon pyruvate, and we lost one of those carbons, and that's where the carbon came from. Carbon dioxide came from. I'm not going to go that detailed. But your book teaches it, so I am going to teach it. So, the remaining molecules, so again, to remind you, we have this, and I'm trying to draw it the way I've shown you in the pictures earlier. We have that 3-carbon molecule, that's pyruvate. We lob one of them off, right? That one goes off to make CO2. And then what we're left with is this 2-carbon molecule that's called acetic acid. For some reason, and the reason I put a star next to that is for some reason your book doesn't even mention this. So if you take notes, definitely write that down. Because that will probably be a test question, even though it's not in your textbook, and I usually don't do that. Which of the 3 stages is associated with acetic acid? And the answer is citric acid cycle. Once again, one of the reasons I don't call the citric acid cycle the Krebs cycle, because any questions you get that ask about acid, they should go together. So just remember, when you hear about citric acid cycle in the exam, I mean, that's it, yeah, that too. You should associate acetic acid with citric acid cycle. Anyway, any questions so far? All right, maybe we can at least get through the citric acid cycle before it's time to go. So in the process of doing this, of losing a carbon to make acetic acid, again, we're stripping away high energy electrons. So there it is again, an NAD plus forming NADH. That's the theme for the first 2 stages. Is all about stripping away high energy electrons. So again, you can see it happening here. And again, I'm not going to get that specific. I'm not going to ask you, how do we strip high energy electrons away in the citric acid cycle? Nope. But you definitely need to remember that citric acid cycle is one of those that's stripping away high energy electrons. Each acetic acid is then attached to a molecule called coenzyme A, abbreviated CoA. So when you bring that acetic acid together and you squish it together with this thing called coenzyme A, then that new molecule is called acetyl-CoA. You do not need to know how that's formed, but you might get a test question that says, and which of the 3 stages is associated with acetyl-CoA? And the answer is the citric acid cycle. So the reason this happens is, again, whatever it is about the acetic acid, it cannot make it into the mitochondria. And you can look that up for independent work if you want. Why can't acetic acid come into the mitochondria? Why does it need to be attached to this coenzyme A? So you can look that up if you want, but you don't need to know it. All you need to know is, technically you don't even need to know that acetic acid is attached to coenzyme A. You just need to know that acetyl-CoA is associated with the citric acid cycle. Yeah, so any questions about this slide? All right, so once again, as a reminder, we're talking about the citric acid cycle, which is the middle of the 3 stages. Citric acid cycle takes the products, well, that's not a good picture. Yeah, the citric acid cycle takes the products of glycolysis, which is pyruvate, and changes it into acetic acid and then acetyl-CoA. But the more important thing here is we're producing a little bit of ATP and we're stripping away high energy electrons. Okay, we can knock this out in 1 minute. So again, the input is pyruvic acid, aka pyruvate. We lob off one of the carbons to form carbon dioxide, then we're left with the 2-carbon molecule called acetic acid. That thing is not ready to go into the mitochondria, so we attach it to coenzyme A. In the process of doing this, more importantly, we're stripping away high energy electrons, and then we're left with acetyl-CoA, and that thing is now ready to go into the citric acid cycle. So technically, everything I taught you is not really in the citric acid cycle. It's preparation for the citric acid cycle. As far as we're concerned in the exam, to keep things simple, this is the citric acid cycle. Right? I mean, and technically, again, this is kind of the in-between glycolysis and citric acid cycle. But as far as we're concerned, this is citric acid cycle. Let's see, I guess the last word for attendance will be, but did I do acid last time? No, okay, good. We'll do acid this time. Acid is the last word for attendance. It's 8:50. You guys have a great weekend. I'm gonna be on office hours only until 10, and then I have meetings. Oh, yeah.